Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Biology Combined Science Paper 1 Higher Tier from 2024. Question number 4 is on the topic of bioenergetics. This question is about photosynthesis. What is the symbol equation for photosynthesis? That should be carbon dioxide plus carbon dioxide plus water to give you glucose and oxygen, which looks like it's a third one. It is balanced as well. A student investigated the relationship between temperature and the rate of photosynthesis. So this is testing you on a required practical. The student measured the volume of gas produced by an aquatic plant in 1 hour. The student collected the gas in a measuring cylinder. Table 2 shows the results. So you've got from temperatures 10 to 45 going up in 5 degrees, and the volume of gas produced in 1 hour starts at 1, 2, 4, 8, 16, 18, 16. So 30 and 35 actually produce the same amount of gas. 40 it went down, and 45 it went to 0. So, sorry, 45 it went to 0. So the optimum temperature is somewhere between 30 and maybe just above 35, because it's still 16 at 35. Okay. Describe the effect of increasing temperature on the rate of photosynthesis. Use data from the table. So overall, as the temperature increases, the rate of photosynthesis is increasing because the amount of gas produced is increasing. But after 30, it stabilizes, and then after 35, it goes down. At 40, it goes down to 0. So if I was to draw a really simple graph, I'm not sure if the question is actually asking you to draw a graph. It's going up to about 30, staying constant at 35, and then it goes down to 0, like that. So let's write the description. As temperature increases, the rate of photosynthesis also increases. The fastest rate is between 0 degrees. Oh, actually, hold on. The fastest rate, if I'm saying the fastest rate, is between 30 and 35. 30 degrees and 35 degrees Celsius. You have to mention and both of these temperatures. So how does this increase? So it's actually doubling, isn't it? So 1 goes to 2, 2 goes to 4, 4 goes to 8, 8 goes to 16. So it's doubling for every 5-degree increase. So up to 35 degrees Celsius, the rate doubles for every 5-degree increase. That will actually give you the 3 marks, but I'm going to mention this point as at 40 degrees, the rate is 0. Or you could say after 35, rate decreases, and at 40, it's 0. Explain why no gas was produced by the plant at 45 degrees. Now, at 45 degrees, the temperature is too hot. So how does that actually affect the rate of photosynthesis? Enzymes in the chloroplast are denatured. You could say destroyed as well. Since the high temperature changes the active site shape. Now, the most important part of the enzyme is the active site. If the active site shape changes, then the substrate can't fit into it. So if that's the enzyme and that's the active site, and this is your substrate, which should be fitting into this, they're complementary. If this active site shape changes, it's destroyed, then this substrate will not fit into there. That means the enzyme is not going to be working anymore effectively, or it won't work at all. How could the student increase the accuracy of the results? Repeat the investigation collecting the gas for 24 hours wouldn't help. Repeat the investigation measuring the volume of gas at 0.5 centimeter cube. I don't think that will help. Repeat the investigation using a different aquatic plant. The best would be to repeat the investigation using temperatures of 5 and 50 degrees. Now, 5 and 50 degrees. Will that help? Actually, let's go back. Repeat the investigation measuring the volume of gas to 0.5 centimeter cube. Looks like it's right, because we know that it won't work at 40 degrees. So there's no point in checking 50 degrees Celsius for the last point. How did they actually do this? Volume of gas produced in 1 hour in centimeter cube. So they're only measuring it in per centimeter cube. So you could actually measure it 0.5 as well. So that would actually increase the accuracy. A person grows tomatoes in a greenhouse. The mean temperature of the greenhouse is 15 degrees Celsius. A heater would keep the temperature of the greenhouse at 25 degrees Celsius. Suggest 2 reasons against using a heater set at 25 degrees in the greenhouse. Now, you would want the temperature to be higher. Do not refer to the cost. Well, if you increase the temperature from 15 to 25, the rate of photosynthesis is going to be higher. If the person is trying to grow tomatoes, that means you're going to have a whole lot of tomatoes as well. So may have too many tomatoes, which could be a problem to eat. Well, if you're trying to sell it, then it should be a problem, isn't it? Although you're increasing the temperature, something else might actually become a limiting factor. So increasing the temperature might not actually have that much of an effect if something else is becoming a limiting factor like carbon dioxide levels or even light. So if it's during the winter time, the amount of sunlight received is going to be less. So having a high temperature is probably not going to help that much. Other factors would limit photosynthesis. Example, light intensity. A person cut a stem from one of the tomato plants. The cut stem was placed in soil to grow new roots. Which tissue in the cut stem will differentiate into new root cells? That would be the meristem. The epidermis is part of the leaf, which is to protect the leaf. The mesophyll, you got the palisade and the spongy, which are mainly to do with photosynthesis. The phloem is to do transport sugars. The meristem is actually found in the tip of a shoot. So if that's the shoot, they are also found in the roots as well. Didn't want that to happen. So let me draw that bigger. If that's the shoot or the root, and this part, this front bit is actually the meristem, the growing part of the shoot and the root. That brings us to the end of the question, and I will see you in question number 5.